Dr. John Brittnacher is going to be giving the next talk, just on a personal note, and there will be more thank yous later. But planning a conference, turns out, is a really stressful, hard thing, full of weird politics and drama that one might not have necessarily anticipated when they decided to throw a fun event like this. And there were times where it was very hard to want to continue. Honestly, we won't get into the depths of all that, but we'll just say that John Brittnacher was the person that I was able to call, and in many ways kind of saved the event at least a couple of times. So thank you first to John for that. Secondly, also, he tirelessly works to keep the ICPS going. He does so much of the work of it, and honestly, that's another thing that probably wouldn't be happening if it wasn't for him. So thank you so much for that as well. One third thing. He did the seed bank. He does the seed bank, the ICPS seed bank, for years and decades even. How long have you done it for? It's so long. A lot of people in here, have you ever made a seed pack? Like folded up little envelopes and put the 10, 20 seeds in there, sent it to a friend or loved one? It's hell. It's hell when you do it 1,000 times.
Yeah, I can even tell you that now with Joe, I send him seeds and I tell him I absolutely cannot do one more.
Yeah, we sold seeds for a while at Calcorn, as you know, and it was so unlucrative, but we stopped because, so thank you for that, too. Okay, also, would you believe that this man made the shirt that he's wearing? He made that shirt. He's really him and Mark are really super into fabric arts and art to wear.
Exactly.
And he made that shirt. Amazing. Applaud for the shirt. He also has a PhD in evolutionary genetics from UC Davis and did a postdoctoral work at University of Wisconsin in Madison. In 2001, he took over the website and then the technical stuff for the ICPS, and as we said, he's done a lot more since then. Seed bank manager. Because his whole life he has been into raising plants. He says that plant propagation is a total obsession for him. I can't live without plants around me. Today we'll be talking about Drosera, filiformis, and friends, and do plants really care about species? John Brittnacher.
Thank you, David. So, why study Drosera filiformis with respect to species?
I can't hear you. Okay, fine.
Oh. The shirt's too nice for this mic. That's the joke. It can be on that side. Try that.
Is this better?
Yes.
Okay. So, why study Drosera filiformis? Well, for one thing, I can grow it. It's a large plant. It grows really well in Southern Oregon, and it has big flowers. So just doing manipulations and stuff like that are very simple. And as I say friends, I actually think it has two friends. There's three of them in the Drosera filiformis complex. And anywhere you have three species at least, and some of them at least can interbreed, you're going to find something interesting. Doesn't matter whether it's a carnivorous plant or not, but you may as well just do it with carnivorous plants. So that's what this talk is about. And I'm not really going to answer that question. I'm going to define what that question is all through the whole thing. And even when I'm done, you probably won't even know why I have that subtitle there. But at least, that's what my whole science career is about. And, in some ways, talking about species and what is a species, that's like the third rail of science. Just don't go there. And that's where I go, because it's so cool. The first part of the talk is a review of formal taxonomy of the Drosera filiformis complex. And some of you probably realize that I have an extreme disdain for taxonomy. And I wanted to tell you how that happened. When I was a grad student, I'd go out and collect flies. I mean, my PhD was working with flies. And I also work with live flies and wild-caught flies. And we'd bring back all these flies, and there would be ones I didn't know what they were, but I knew they were in the same genus as the ones I had. But they also had a lot of sibling species, they call them, which are so closely related, I mean, they can't interbreed or anything, but they're close enough related that they're hard to key out. And so I'd bring the flies in and go, okay, what are you? And then I try to key them. And I go through, and, how when you do a key, you try to figure out, because it's all what all the words mean and look stuff up. And I'd key them out, and I'd get stuck on the section where it would say, scutal bristles are convergent or divergent. And I'd look at my flies, they were parallel. And so I started figuring this stuff out, trying to go back to what's going on here. And then I realized that, the keys only work with dead, dried flies. It's like, I don't work with dead, dried flies. But I had a lot of friends that were animal or insect taxonomists. So they set me up, taught me how to point them up and pin them down, and all this kind of stuff. The whole accoutrement. They gave me everything, beautiful cedar box to keep everything in and all that kind of stuff. So I did it. And then, after they dried out, guess what? I don't remember actually if they came in or came out, but they did move. And okay, now I know what species I'm working with. It's dead. I don't work with dead stuff. So, right when you're in the beginning of your career, you all of a sudden are faced with this roadblock and this what? Why can't you just give me something I can use? So that's why I'm there. Okay, so there'll be 4 parts in this talk. So that will be the formal taxonomy. I try to be a good boy. As you will see I'm going to have an article on this. So part of what you see here will be an article in CPN, September CPN. It'll be the 3rd section, actually. I'm really good in that until the last paragraph, but I won't tell you what it says. But you'll figure it out here because it really says it. I'll say it here today too. But really, we do need that formal taxonomy. So I want to be fair. So next we're going to do some tours. New Jersey, North Carolina, and Florida. Most of that work is relying on Jason Sepka. And most of the slides are his, except for North Carolina. And I have 1 day out in the field to prove that I have been out in the field with Drosera filiformis. But really, to study these plants, you have to do you have to be out in the field with them constantly, especially during the time they're blooming. So you have to be in Florida at the absolute worst time of the year, out there every morning with the sunrise, and then be wandering around checking out all these plants, seeing what they're doing. It's like, no, I don't go there. Okay, what will be in the September CPN is the photos close up, and I'm going to have way more photos than I'm going to show today. And finally, the 4th part is finally using my genetics. And what's really cool now is they've actually started to figure out how all this stuff works. So we'll get into that, and there's some interesting stuff there. But again, when you're working with these plants, I'm doing the research. If I tell you how many years it is, it's like that doesn't give you an indication of what I did, because life happens. Some years nothing happened on the research. So, but to get into it, so here are the taxonomic names. The first one, filiformis. So when I say Drosera filiformis, that's what I'm referring to. And that's the original species that was done in 1808. When I say Drosera floridana, I mean, but I don't mean filiformis var floridana. I really do mean Drosera floridana. But I don't really argue too much with Barry about calling it a variety, because I'm not even sure he has enough characters to call it a variety. But I'm absolutely convinced it's another species. And then we have Drosera tracei, which was named in 1914. There's nothing wrong with that species description. There's no reason why people shouldn't call it that based on anything in taxonomy. It's just whether you want to believe certain people or other. Now, and then we have Drosera X californica. That's a really unfortunate name, but that's really based on the California sunset. So when I talk about California sunset, that's what I'm referring to. I'll have more clones in the paper. And then Drosera californica var arenarius, one that Barry just named, which is the hybrid between floridana and tracei. Yeah, and California sunset is filiformis and tracei. Now, technically, if floridana was a species, then it wouldn't be X californica. It would have to be something else. And because I don't like that X there, I'm just going to forget that it's there. It's not required to be there according to the rules. But you either use it or you don't use it, okay? So you can't say X californica is one thing and not X californica is something else. And I really need to thank Fernando for that, because at one point I had him, because like all my articles on the ICPS website are reviewed. And I usually have Andreas and Fernando look at them to make sure I'm not saying things that are bad. And Fernando just said, just relax. I mean, just don't even fight it, and just go one way or the other. Whatever you do, just pick it, choose it, do it. And then finally, Drosera hybrid, that's really only there, well, you could call it part of the species complex, because that's a hybrid between filiformis and intermedia. But that's really what got me interested in this group in the first place. That's a really cool plant. So what is formal taxonomy? This is what formal taxonomy is. Just like formal taxonomy in insects, formal taxonomy in plants is all about dead plants. And like I said, I don't do dead plants, I don't do dead insects. So if this talk was really about formal taxonomy, we'd spend the next 40 minutes on these things. And I'd be showing you tables of measurements and all that kind of stuff. What is interesting is this is what I believe is the replacement type. It's in the images of a website that gives type specimens only, and that's the oldest, that's the one that came up when I was searching for it. And because it was 1817, which was after it was named, and there really aren't any annotations, so we know nothing about it. I mean, they could just as easily have been from cultivated plants. This again is lectotype, which is also, that's a replacement type, correct? Yeah, so they didn't even have one of the original one. But it's just so cool to look here and just see who touched this thing. And if you go to online, a lot of this type specimen, or the other specimens, herbarium specimens, it's just, I mean, I just always thought it was so cool because you pull up the images and you look at it, and right down here you'll see a little sticker, and it has a name, and it says L. Mellenchamp. And and it's all, a lot of them, there's even more stickers from other people, and they're all changing the name this way and that way. And, I mean, it's just the history. I mean, so I really love that part of this, because you can research the history of the names, history of who's touched the specimens, how people think about it. Now, these are brand new herbarium sheets that Barry made on his new names. And so this is Drosera marinara. Those are big plants. I mean, look at this. That's incredible. But then I have real problems with this one. I mean, I've grown Floridana a lot. And it was the last batch of seeds I got, which came from Atlanta Botanical Garden. And they're F1s from seeds that they collected, and I know if it's from Atlanta, they had permits to do it, or permission. And when I got it, it says, oh, these are really cool plants. I mean, really spectacular Floridana plants. And it's like, I started growing them out and looking at the flowers and going, I don't think so. They're just, the plants are too big. They're bigger than any I've ever grown before of Floridanas. The flowers are bigger than any Floridana flowers I'd seen before. And it's like, I think those are hybrids. That is more what I expect. But then again, knowing Barry, he probably used this one as the type specimen. Well, that's the way it goes. Okay, this, when I found this, I can't believe how happy I was. I mean, it was just, that's the type specimen for Drosera hybrida. When I was doing my first paper on hybrida, no one knew where that specimen was. I mean, I don't remember if it was Andreas or Jan who told me that chances are it was lost during World War II because it was in Berlin. It was in Berlin, but it wasn't lost. So that means that John McFarlane mounted that plant that he dug up in New Jersey when he dug up every single one of them. So, and then because California Sunset is a cultivar, now we're dealing with live plants again. That's why when people ask me, should I do it as formal species thing or should I do a cultivar? I said, do it as a cultivar. I mean, then you can be talking about a real plant and have real pictures. And it's easy too. You just write it up, making sure you have certain information in it. Send it off to Bob Zemer, and Bob will then also send it off to Jan to verify everything's okay. And you're done. It'd be better if you had a nicer photo of that, but this is so old. I mean, as CPN, at that time, CPN, the photos were approximately like that anyways. So this is formal taxonomy, but I'm not going to force you to look at any more. Oh, actually, there's going to be one more set of herbarium sheets, but that's later. Okay, so next we're on to the tour. This is the first site we're going to look at. This is in Ocean County, New Jersey. This is one of the most spectacular photos that that Jason has, showing filiformis growing. But it's like, when I see things like that and I see the trees around this, I wonder what's special about this site. Why is there so much filiformis? And so Jason told me where it's at. I won't tell you exactly, but it's over here somewhere. It's in in this area where you see all these, these lines going like this and around in circles and stuff like that. Well, those are ATV tracks. So people go out there and just drive their ATVs like crazy. They disturb the habitat, and the filiformis just go crazy on the edges. And in fact, most of the filiformis that Jason has shown me or told me about, they're all in disturbed habitats. I mean, that's really all that's left of areas. It's sort of like if you go in and plant all these trees, or if you don't burn the trees, or if you don't cut the trees, they're going to lose their habitat. I mean, it's like you go and you want to preserve them, and by preserving them, you kill them. But then you have to expose them to ATVs and all that kind of stuff. But you see these aren't natural here. This whole site was a silica sand gravel mine, I presume for making glass, because I know of other sites that were for making glass, but I don't know specifically about this one. But you can see that this is all going to be subdivided for homes. Now, of course, everybody who lives here is suing the company that wants to do this because they're worried about their water quality and water availability once you start getting so many homes out in the pine barrens. And Jason's afraid that once they start sucking out the water, that he's going to lose that site. But here it is. I mean, you can enjoy it while you see it. So you can see all those gravel, the big rounded thing. That's all pure silica sand or whatever, granite type stuff. Gorgeous plants. But also notice that they're growing aquatically. I always think that, I should be able to do that. But when they, if you have them in too short of pots, they don't put out any roots. I mean, one problem I have growing these guys, not so much filiformis, but like the hybridas, is it's really wet in our spring when they start growing. And all of a sudden we go from days in the 60s to immediately to days in the 80s. And it's like, oh, I forgot to grow roots. And they turn all red and go, it's just whatever. So I always think I should be able to grow and then get nicer looking plants, but then I realize, well, if anything happens, they're toast. So again, here's another ATV road somewhere else. But this site has filiformis, intermedia, and hybrida. The hybrida is right here. This is from Jason's paper in I think it's the last CP, I don't know, before last. But you can see again what kind of site we're talking about. And then when Jason found this site, we were so excited because it's actually the largest area or site that he found with hybrida. Unfortunately, the photos are not that clear. But you can see there are some, like I think this is whoops, keep missing the button. Here's I think that's a filiformis there. But this is like the best clump right here that I could find in the photo of hybrida. Again, we're on a road. And I thought, oh great, we finally found the tetraploids. Because I always bug him every year to go out and look for tetraploids. It turned out, no. There was some guy that would come through every year who wasn't supposed to do it, but he did, and mowed it. Talk about leaf cuttings. But he also, when he mowed off all of the scapes, so like we couldn't check for seeds. And then the next year, probably because he was pissed off, but he's a volunteer. What can you do with volunteers? He bushwhacked all these guys out to preserve the site, right? Then a hurricane came through and washed sand over the whole site. So Jason was out there on his hands and knees digging out the plants after that happened. Yeah. No, Jason only lives for being out in the field.
Just one quick story about Jason.
Okay, sure.
So Jason Sepka, he's like, they call him Nature Boy.
Yeah.
And there was he lives in New Jersey area, and there was a purpurea bog. A lot of people don't know this, but the beavers would come in and they'll build a dam in the seepage, and then it was flooding this purpurea bog, and they were all going to die. And so he went out there and camped, and he came into the nursery he was working at the next day, and slapped the beaver tail down on the table, because he had shot the beaver and eaten it, camping there overnight, probably all barefoot the entire time.
And then, okay, so this is a close-up of the hybrida, and what's also curious here is you can see false vivipary, and that's another way that they can spread locally. That there's a site in northern Oregon in the mountains where there's just a huge amount of obovata, I think it's obovata, hybrids. And it's massive. And I asked Barry, well, can you look at the seed for me? Yeah, and he says, everyone he checks, there's no seeds. And I realized, well, if they do, some years they only do false vivipary. And they just, could eventually fall over and root. So, all these, it's sort of like, I always think that I should be able to, because, you get taught that it's going to happen. They're going to make tetraploids. And I think, but we're not finding any. And then it gets planted out in Butterfly Valley, and it's like, it turns into tetraploid. I mean, it's what is it? I mean, how does that all work? And I don't know. Okay, so now we go down to Columbus County, North Carolina. That's Mark Todd and Victor Holland. And Mark took me to every site in North Carolina where he had ever seen Drosera filiformis. All but one of them were roadside ditches, and the other one was in a state park. We went to 3 of them. Well, the state park, let's just not even go there. And the roadside ditches, they had all kinds of cool stuff, but no filiformis. And this is a little off topic, but I mean, this side was really, it wasn't really that great out here. What was really cool was back here, in the shade, in the drainage ditch, were Sarracenia minor. And they're just all tall and floppy because they're in the shade. And on this side of the road was Sarracenia venosa. All in the ditch. I mean, I don't even have a photo because it's like, I just see this little picture hanging out there. But there's none of those species out here in the main part of the bog. You get, here's a little flava, and this is the actual bog. It's, looking down like the bog. So it's just under this power line, power line easement. I don't really know why there's all these tire tracks here, and you can actually see evidence that they drove through here. And, if I'm kind, I will think that it's because the power company came through bushwhacking or something like that. But I tend not to be kind. I think it's where guys come to get their trucks muddy. So this is what the sundews were like there. So you can see the filiformis, intermediate and capillaris just sort of spread out through here. So that was my first time seeing filiformis live in the wild. This next site, which when I look at my photos, it's like, wait a minute, that's the other site. No, it is. It's this site because it's different in the background. It's like, and this is it. These are the 2 sites. They're half a mile apart under the same power lines. They get sprayed by herbicides and bushwhacked and all that, both of them at the same time when that happens. Now, I don't know how often they're going to do it, whether they do it again. Mark said that they're not so much bushwhacking anymore, they're just going in and spraying. And again, it's got this water back here and there's all these tire tracks through it. But it's got some nice flowers. And you can sort of see some of the filiformis, like here's a filiformis scape. And right now you can notice that scape's almost bloomed out. You can see a little intermedia down here. So one of the things I wanted to find out about was, why aren't there any hybridas there? Or can there be, or could there be hybridas there? Well, the intermedias, they're not even thinking about blooming yet. So just right there, unless you've got plants that are blooming out of sync, which they're very unlikely to do or want to do, let's put it that way, there may be some way they could be induced to do it. There's just no way it's going to happen. But then also, and this is just another one you can just maybe see a little bit better that they're almost, here's escape and it's almost bloomed out. And then there's one more plant that was actually the best one I could photograph, this one here. And those are big plants. Now, since I hadn't seen them in New Jersey, I really couldn't tell myself. And they were really red. But then, once my focus got off of this, I thought, well, what's this? So here's this Astropurpurea. And it's like, but wait a minute, there's nothing even close to that in that area. I mean, in that set of plants there, the flowers. I mean, there were a couple of Cupreas, but most of them were regular Regalias. Then I realized, oh, yeah, shovel holes. So that's your fate. I mean, people say, oh, Astropurpureas are rare in North Carolina, in the Carolinas. Well, I don't want to make any accusations here, but whatever. And then there was one more site that Todd didn't know. We had to drop him off because it was getting late, and we had to drive halfway back to Durham to get to this site. When I saw this sheet, I really got excited. But that's before I'd been there, because this plant was just huge. And then look at these scapes. It's like I've never seen grown a filiformis that looked like that. It wasn't until like a couple weeks ago that I found this one, too, from the same site. That's collected at the same time. The collector sent sheets off the different herbariums, so they would have, multiple herbariums would have sheets from the same site. So it's like, I got to go there. I mean, I really have to see, because this just looks so much like a hyron. Well, we got there. That's what we found. So there's this area of death. And I really felt uncomfortable taking photos of what's upstream from this, just because it had people's houses, and it had the most beautiful cornfield you've ever seen. There wasn't a weed in sight, and all the plants, just perfect and the same height, and all that kind of.
Well fertilized?
No, I don't think it's, well, can you say atrazine? I don't know if it's atrazine or not. It's just I showed the photos to somebody, and he said atrazine, which is an herbicide that they use because now they can sell that corn as non-GMO, whereas if they use GMO corn and they use something safer like glyphosate, this wouldn't have happened. But North Carolina is one of the few states that allows atrazine. So whether it's atrazine or not, I don't know, but it's one of the few states that actually allows them to use it. But you could just follow this trail of death from that cornfield across to a culvert that went underneath the road. Down the other side of where it went underneath the road, it was just full of shrubs and just totally choked. Into this area, it was like a holding area. Then it went under the railroad tracks, and this was the other side. And you could just follow this trail of death until we couldn't see any further into this thicket here. And it's like, not so much the filiformis, so, I mean, the filiformis may not have been there before this, but you do this to a wetland for how much money? But that's North Carolina. So let's go out to Florida. So these again, we're back to Jason's photos. So here's, I don't know how typical this, most of you probably, more of you have probably been, I've never been there, and there's no way. So here you got filiformis growing, Tracei growing in with the flowers in a real grass. I mean, this doesn't look anything like any of the previous photos I showed you. Look more close up. And another one close up. I mean, who could say this is the same species as Tracei? As filiformis. And then we can also go to a Floridana site. I'm not sure which site. There's two major sites. One of them is just, as far as I can tell from talking, looking at photos and stuff, is a huge hybrid swarm. But that's what it looks like. I mean, it's growing on almost pure white sand. I think that's why they're so red. Just you think about the heat generated off this sand. Really cool. And then that's one in a little shadier area, and they're not quite as dark. And finally, a bright race photo, and then here's the arid area. So it is, very different. It looks very different from that. But you get into hybrid swarms, you get back crosses to Drosera, you would get back crosses to Floridana. So you just expect a big mess once you start getting these things mixing up. And my fear for this, the biggest of the sites is not fear per se, because it's really cool scientifically, is you're getting these hybrids between Floridana and Drosera, and it's just a cool example of a species potentially, possibly in the future, going extinct by hybridization. Now, I'm not horrified by that because, like Fernando was saying, I mean, this has been going on forever. I mean, we're seeing just a little tiny time slice of things that have been going on for thousands of years. I mean, going back to New Jersey, 20,000 years ago, the sites where I was showing those photos, that was tundra. Filiformis could not have been there. Where was it? And the New Jersey and the North Carolina plants are sufficiently different that you know that if there was a refuge, they were not in the same one. Alex Iltz has a really, really cool video on what's called glacial stragglers, but it's about a refuge in Florida, which is actually the epicenter also of Sarracenia. And plants that got stuck in the refuge is mostly what it's about, but he talks about it. It's got a lot of, it's nice. So now we're done with that. So now we're on to cultivated plants. So these are my plants grown in Ashland. So I just went out, grabbed a few, lined them up, and Drosera, this is like, I think I did this mid-June last year. Drosera really, I don't remember if this plant had been outside all winter or not. I don't think so because of the way it looks right there. It's really not starting to, it's just recovering from the winter. So it eventually got, like you expect Drosera to be with much longer leaves and falling all over the place. And then this is the New Jersey filiformis. See how red it is compared to North Carolina. The North Carolina plants tend to be a little bit bigger and a little bit paler. And then this is the California sunset, which is normally intermediate between these two. Like I said, this one's just getting started. What's really cool was these were really too small to bloom in the spring. Around August, I think, this one set up a scape, and we'll get more into that later. It actually bloomed after all of, filiformis bloomed. Because normally they're the first thing to bloom. I mean, when I put them in my garage, they bloom in March. And then this is my favorite Floridana clone. I got it from Jason. It has white flowers. It's much rarer than anyone I've ever seen. It tends to be a little bit shorter, but it's one of those things, it's totally winter hardy in Ashland. And in fact, if I try to protect it, I lose more than if I left them outside totally unprotected, which is bizarre. I mean, because every other Floridana clone I've grown, my favorite is Peter's old one. I mean, you could grow it in a terrarium and it'll never go dormant. And, but if you give it frost, it's dead. And most of the ones that I have grown from the seed bank, because I always like to make sure, it's like, oh, this is cool. I wonder if it really is what it says it is, and I wonder, you know, if people are going to be able to germinate it, that they've all been the same until I got these ones from ABG. So that's why I feel that way about that plant, because I've grown so many others that were just absolutely different. And then, so these are some of the close-ups. So this is from California Carnivorous, your Bruce plant. To me, that is sort of like the Tracei. It's the most extreme. It doesn't do anything weird. And it's pretty cool. Whereas, if this is a problem, you start looking at herbarium sheets, and you start seeing a lot of plants that deviate from this. But I think like in Drosera of the world, Barry picked a plant that I now think is a hybrid to represent Tracei from one of my photos. This is the one he should have used. And then this is, you know, California Sunset. It's sort of intermediate with the Ocean County filiformis. And then finally, this is that white flower Floridana. And in Barry's monograph in CPN on filiformis and Tracei, I mean, I don't think he'd be embarrassed if I called it the rice index, but I do. That what he does is he goes to old herbarium sheets, and you know how it is when you got dried plants, how the tentacles do all weird things. And so he tries to find ones that didn't, that are sticking out straight, and he measures how long they are, and then he measures, so like you would just, like let's say here, and then measure across the distance right there. And then you make a ratio of tentacle length to leaf width, and you get an index for that leaf. And so he did that across all kinds of these herbarium sheets, and came up with a range of 0.9 to 0.23. This is the 1 sigma, which means that there were outliers that were even more extreme. But like I said, when I look at some of these specimens he probably himself measured, it's like there's always a leaf or two that looks like this. It's not with the red, but I meant the tentacles in the leaf are like that. And they're usually like the first or second leaves of the spring. And like I have absolutely no idea what to think about that. But you know, this plant doesn't do that. The ones that Mike Wilder sent me from Hammer Ranch doesn't do that. So yeah, and then as you expect, you know, the California Sunset is in between. And then Barry got this set of numbers for filiformis, but he also lumped in the Floridana with the filiformis, so you really can't take that. You have to know there's some, you know, he just lumped them. So, but you can also see that the numbers I get from these photos, I mean, here I'm talking about, I go out, I bring the plant in, photo it, and I just, you know, I can't be photoing it. I mean, it's just, you can only do so many. So I just take a leaf that I think is pretty much representative of what's there, unless there really is something I think I need to photo too, but at least one that's representative. And I don't get very many numbers when I do that. But the cool part is that I can just take my little ruler and display this on my screen, on my computer, and measure stuff and do the ratio. I mean, the ratio is independent of units of measure. So it's just something that's really quick, easy, and you can also, if you can get decent herbarium sheets, go back and get your own numbers off those herbarium sheets too. So, but I think that, I haven't done an experiment. I mean, I think if you took this out and dried it, that the tentacles won't shrink, but the lamina will, so, which is why his numbers don't, like my results don't fit within his set of numbers. So then I also photoed the flowers. Now these images are not to scale. See, the other ones with the leaves, I can set my camera to the minimum as close as it can be with the tentacles that are closest to the camera. And then the camera I use is controlled by my computer to take multiple photos, so I use photo stacking. So I know that, so I set the camera the same way all the time. It's the same distance from the leaf, and I know that they're all the same. But I didn't do that for the flowers. Because the flowers is a different size, and I really want to get as much detail in the photos as I can. So this is like a typical Drosera flower. This is the California sunset, which I don't know why the petals are so wide. The Florida giant is the one with the wide petals. So this is California sunset. Here's Florida giant from Damon. And I don't know why these are so wide, but then I see this down here, which we'll get to in a minute. That could be it. And then this is the New Jersey filiformis. It looks same form factor, but it's smaller than the Drosera. But see, then here's Columbus County, North Carolina, and it's got, sort of flowers like that, and flowers like this. And I thought, oh, cool. I mean, Damon just mixed his plants up. But like, no, I don't think so. I mean, it's just like, what do you? So, but this is interesting. So the flowers from Columbus County are larger than New Jersey. The petal shape is different. There's also, the stamens can look a little different. But the main thing you can tell, between filiformis and Drosera, but see, these already started to bend in a bit, is most of the Drosera flowers, especially like you can see here, the stamens are too long. The plants do not self-pollinate or pollinate very well. Whereas the New Jersey ones, they bend in and self-pollinate like crazy. I mean, all the filiformis do that. And then these are the two major clones I have of Floridana. This is the white flower, and then this is what I call my Peter clone. And then this is one of the plants that I got from the ABG group. It's huge. I mean, it's a much bigger flower. The stamens, the stigmas are, whatever. The female parts are really long. So can you say hybrid? Okay, and then we're on hybrids. So here, these actually, from a couple of weeks ago. This is the white flower plant, which this year did spectacularly outside. These are my North Carolina plants, some of them, and then intermediate. So these are, like all the pots of intermediate I have, you can only see I've really got flowers on a couple scapes. So it's a real race. I'm sitting out there, telling the intermediate to get going. I try to do things to get it to bloom sooner. But the bloom overlap, it's like 1 to 2 weeks in my conditions between the intermediate and the filiformis. And the little green threads are crosses between the filiformis and the intermediate, and I do them both directions. And that's what I found, I talked yesterday about flowers, so amusing because, I have to set my phone to go off at 10:15 in the morning to go out and pollinate the flowers, because these guys are just opening at 10 o'clock. And these guys start closing at about 10:30. So, and there's a couple days where we had, the day we had thunderstorms that started all the fires. I had to take this guy into the house, put him like with one of my LED spotlights almost right up to the flowers to keep the flowers open long enough so I could actually do some crosses, do all that stuff. And then when we got smoky days, really smoky days, I mean days where you couldn't see a block because there's so much smoke, these guys didn't even bother opening at all. And and here it's like, this is in my window when I'm doing my research, and the flowers weren't even open for me. And so I really, feel for people who want to do this because, and you notice I haven't actually done anything with this guy yet. And the thing is that I did do the cross last year with this is the female. I got these really strange plants out of it. I haven't taken a photo of them yet. And I can't tell if they're just weird plants of this, from this, or if they actually were hybrids or not. So you have to be really careful when you do these crosses. People post on there and said, I did this cross and this is what I got out, and so it's a hybrid. You've got to be able to prove that you've got a hybrid. And if you don't do the crosses right, you can't be sure until they either get bigger, you can see the flowers. You have to have some confirmation. And with these, it's sort of like, well, I have to wait till those little tiny runty things bloom, if they ever do, because Irish Schneider sent me some of his hybrids. They're never going to bloom. I can't confirm anything with them. They don't even grow, some of them. So, but now, now that this guy stopped blooming, then I started doing crosses in this direction. But I also try to do it both ways because some of these crosses don't work both ways. But these are ones that, these were already been published in the CPN, but I'm just showing you here for people who didn't see it. So this is new hybridus. And I can also do intermediate by California Sunset. I think I can do these hybridus both directions, but I'm not 100% sure. I think I did the California Sunset both directions, but I only got a couple plants. I got plants in this direction, but I'm not sure what happened with the other. Because the problem is too, when you're working with intermediate flowers, you gotta pull the stamens off without getting pollen all over the flower. And because it's so dry, I'm not going to pull those petals off. Because it really needs the petals to be in there and keep things from drying out before they can, do what they do. So there isn't a whole lot of pollen you can get off an intermediate to pollinate a much larger flower, although I can get plenty of pollen going the other way. But the other thing is that try to pull those stamens off without pulling the stigmas off too. So here you go and you go, oh, okay, I think I've got it now. Pull it up and go, oh shit. That was your flower, the flower you had open, So, and again, this is Floridana by Intermedia. And that plant this year, they were really nice. I'm sort of afraid to put them outside over the winter because it's Peter's plant by Intermedia. Now I'm trying to get the white flower by Intermedia, so those can be outdoor plants. And then this is the filiformis North Carolina by Intermedia plant. I have no idea what to make of this. It is really bizarre.
You grow that?
Huh? Do I grow? That is what I have. That's one plant that's made multiple growth points. The flowers, the leaves so far have never opened enough to even see if they have rounded tips. But the tentacles are red, they're full length, so it's sort of like, well, that's an intermediate character. So we'll see what happens. It's a really nasty thing, though. In a big photo blown up, yes, but in person. So what doesn't work? So from that one Tracy escape that I did the crosses with Intermedia, with Tracy is the female, the capsule swelled up and then died. And on intermedia as the female parent, I got hollow C's. And it's like, cool, I finally found something I wanted to find. Or not wanted to find, but I found something weird. Me always wanting something weird. So, also pointing out that you already saw the filiformis by Tracii, then by intermedia. That's California sunset by intermedia. I have those. So something screwy is going on between intermedia and Tracii that doesn't happen with filiformis. So I started looking this stuff up, and I mean, this really smells like epigenetic signaling failure, which means that the whole concept of this is that there's no difference in the genes. Both plants have the same genes. But the epigenetics are the proteins and RNA complexes that tell the genome what to turn on and what not to turn on. And I mean, now we're really going to get geeky. What happens is that under certain conditions, you get a lot of genomic reorganization. There's transposable elements. You get a transposable element breakout. Now, it's not like you're going to go in and the plants are going to have huge, things going on that's going to make them sterile. I mean, these sorts of breakouts can happen over hundreds of years at a very slow rate, but any genetic changes that are going to happen in the genome are happening orders of magnitude faster than they would have otherwise happen. And what happens is that the plant has to get those transposable elements back under control, and then it has to get all its signaling for turning genes on and off corrected. And what you end up with is a plant that, again, is identical to what you started with, except the genome's all scrambled. And this is why I think reproductive isolation is totally inappropriate or does not pertain to species at all. Because you can get that happening within the species. There's hints that, for instance, I can't remember its name. The plant that looks like spatulata that lives in Texas and Ribifolia. Ivan Schneider, that he's made crosses within the species and they don't work. That's the only evidence I have at this point. Yeah. Mm-hmm. That's what Ivan says that. I haven't confirmed it. I haven't seen anything scientifically done or anything like that. But that's the sort of thing you do expect to happen. And there's nothing in respect to adaptations for different leaf structure or anything like that that causes the genetic reorganization. That can just happen by normal gene changes. But most of what you get with respect to sterility and crosses has to do with genomic rearrangements. So you're getting into an area where, yes, there's a huge body of research done on development of genetic isolation, by changing flower type colors and types and stuff like that. But underlying all this is something that is not Darwinian type evolution. It's just what happens. It's what, and I say in my pages on the ICPS website, I mean, we wouldn't be here if this didn't happen because evolution on its own cannot operate fast enough because you really want to keep your mutation rate at a minimum, but the lower your mutation rate is, that's your low potential evolution rate. And there's times when you really need to move fast. And usually those things, at least in animals, get triggered when the animal is stressed. I think in plants it happens when you get sort of hybrids, or even hybrids. And that hybrid genome that caused the breakout may be totally selected out, but the breakout's happening. And like I said, this goes, when they try to check rates on this, it's not fast. It's, you know, it's hard to demonstrate. So it's not like there's going to be a big field, a group of people studying all this stuff because, well, they got to get their papers out and they have to get their grants. I mean, and it's not really simple. But, and then to go back and also explain why seed capsules and all that. Okay, the seed capsule, the cells for the seed capsule come from the female parent. Those are female parent cells. And you get, the embryo releases, I think, small RNAs, but I'm not 100% sure, that tell those cells to make the seed coat. Then after the seed coat happens, then the embryo starts doing dividing, getting ready to do certain, and once it gets to a certain stage, it does an epigenetic shift. And it's the epigenetic shift which fails because they have to get both parents, actually a lot of it happens in the endosperm, we have triploids. You have to get all three of those chromosomes running full bore to get enough of whatever it is that makes all the nutrients to come into the cell so that the cell can build up the endosperm and the embryo. And if that doesn't happen, you get hollow seeds. So, to me, that's really cool because we're looking at a difference between Drosera and filiformis. We're looking at evolution or result of evolution in the sense of non-Darwinian type evolution happening in these plants. So it would make a nice set of, like, you know, it might have some interesting things that people can study in the future. And then this was just telling you about the course I already told you about, you know, because of that last year and what we're doing. But this is my total of how many flowers I can do this year. And so, like, if I'm not here, those 2 weeks, when I can do these grasses, that's a whole year lost. And that's how many flowers I can get. Pollinated. So, but like even if we can show that North Carolina plants are different from the New Jersey plants, there are absolutely no characteristics between them, as far as I know, other than that little bit of a flower petal or something like that. It would merit giving it kind of a special name. It's just, and see, that's the sad part, is that, you know, even if your only interest is doing science, losing plants just eliminates your ability to study how plants work. If you start dropping out all the species, or just a selection of species, you watch it go, it's sort of like if Fernando had all of a sudden decided that he couldn't use half the species for his study on Drosera. What would that look like? And what would you know? What would you lose? You know, so that's what happened. So, to summarize then, you know, ovarian sheets are just dead dry plants. But yeah, you have to have names. You have to be able to know what you're talking about. You know, there really is a need for that. But it's impossible for ovarian sheets to actually demonstrate or show or be in any way useful for complicated evolutionary genetic things, because there's so many things out there that you really want to name for something, but there really aren't any characters there. And the ones that are most interesting to us, to me, are the ones that haven't been separated from their partner species long enough to have a set of characters that a taxonomist would accept. I mean, you can actually show, I mean, you know, you're someone who, you know, he named a species because it was totally reproductively isolated from
it's parent species. The taxonomist says, well, no, I mean, there aren't enough characters. So when we look at your sheets, we don't know which is which. We can't decide between the two. I mean, I think of species names as, well, that's just the index where you file stuff in the area.
Yeah.
So, but then again, I use the names. And again, when you look at plants in the wild, you're just seeing a snapshot. I mean, that's the thing about what I really find cool about like West Coast flora, is that so many plants live where they could not have lived 15,000 years ago. And just studying how they moved around is, I mean, that's a piece of work right there. Identifying where the refuges were, so on. And what are those plants going to do now when climate changes and they can't move? I like growing plants side by side, so you can really, because when you're out in the field, you're seeing how plants respond to that environment. But when you grow side by side, you can get more of a feel for how they are alike and different. And it's sort of like, I really want to live with my research subjects. I really want to know just an innate sense of what they're like, how they grow. And the only way to do that is to live with them and grow them yourself. And then, I like doing the crosses. I mean, yes, with Sarracenia, I just do crosses like crazy just because I want to create plants. But with the Sundews, it's like, I really want to know how these things work. And like I said at the beginning, any group of species, you're going to find probably a lot of the same things that I find in here. But you can't go very well. Try to grow in South America. Try to grow some of the, and propagate some of the South African very, well enough. And then just little nasty things. The bigger side is really cool. So this is actually my slide. So that's pretty much it. I mean, this is actually taken a botanical part. To give you an idea of this stuff is going to end up. So that's North Carolina, with their collection of the North Carolina filiformis. And I hope they don't better than North Carolina. Those other sites that host, this is it. Thank you.